Welcome, everyone. Today it's my great pleasure to introduce Dr. Jean Gotman, our speaker. Dr. Jean Gotman obtained a degree in electrical engineering from the University of Paris, a master's degree in computer science from Dartmouth College, and a Ph.D. in neuroscience from McGill University. He joined the faculty of the Montreal Neurological Institute of McGill in 1977 and became a full professor in 1993. He pioneered the automatic detection of spikes and seizures in the EEG and made his methods widely available through Stellate, a company that he created in 1986, which developed and sold all over the world equipment and software for EEG, long-term monitoring, and the polysonography. He published 280 peer-reviewed papers and 40 chapters. His research interests include analysis of the EEG, mechanism of the epileptogenesis, seizure generation, and seizure spread in humans, high-frequency oscillation and functional imaging in the diagnosis and study of epilepsy. He received a Research Recognition Award from AES, the Pierre Flohr Award for the ACNS, the Penfield Award of the Canadian League Against Epilepsy, was named Ambassador for Epilepsy by LIAE, and gave the Lennox Lombroso lecture at the AES. So without further delay, Dr. Gotman.
Thank you very much. Thank you for this kind introduction. You can hear me, yeah? Okay. It's a great pleasure to be here in Washington. I particularly appreciate the welcoming weather, which makes me feel right at home. I thought I would see a little spring, but it's the flowers that I saw yesterday are now disappeared. So I will talk to you about how we have been using the combined EEG and fMRI method to localize epileptic foci. I think it's a very promising method, and I would like to share some of its advantages with you. First, we have a few things to say. This is related to the rounds here, the activities at George Washington University. These are the disclosures. I don't have any disclosures. Learning objectives are for you to become familiar with the rationale and the method for performing combined EEG fMRI studies in patients with epilepsy, to understand the type of information that can be gained from this method in the localization of epileptic foci, and finally, to understand what I think is an interesting concept, which is that of the spike onset zone, and now it relate to the seizure onset zone. This is related to accreditation and CME credit. I'll go quickly through this. This is to claiming your CME credit. I think there will be more information about that at the end of the talk. So this is the outline of my talk. First, how do we go about combining EEG and fMRI? And then we'll see some examples of this use in focal epilepsy. And then we'll discuss this, what I think is an interesting concept, that of the spike onset zone, and how it relates to what we're really interested in, which is the seizure onset zone. If we have some time left, we'll talk about epilepsy-related network and then conclude. So, why combine EEG and fMRI? And let's look at some of the major characteristics of EEG. EEG has the great advantage that it reflects neuronal activity directly. In other words, what we see in the EEG is generated by neurons. Of course, we have in the EEG patterns that are specific to epilepsy. In this case, we're interested in the interictal activity in particular, the spikes. The spatial resolution of the EEG is not great. The temporal resolution, of course, is very good. And the EEG is not very sensitive to deep brain regions. Now, the fMRI does not reflect neuronal activity directly. It reflects through cerebrovascular coupling. And I'll spend a minute to go through that for those of you who are less familiar with fMRI. fMRI measures the functional activation of a brain region. And this is what happens when we have neuronal activity. We have neuronal activity increase somewhere. That is followed by an increase in the quantity of deoxyhemoglobin in venous blood as a result of an increased oxygen metabolism in neurons. That is followed by a large increase in blood flow and blood volume, and that in turn is this combination results in a decreased concentration of deoxyhemoglobin in blood because there is such a large increase in blood flow and blood volume, even though there is an increase in the quantity of deoxyhemoglobin. And that's the bold effect, which can be measured by MRI because deoxyhemoglobin is paramagnetic, and less deoxyhemoglobin results in interference with magnetization. So that's the bold effect. This is the time course of the bold effect, and this is so-called the hemodynamic response function. If you have neuronal activation here at time 0, then you have a peak bold response about 5 or 6 seconds after, you have an undershoot 12 seconds later, and a 20 second after that you return to baseline. So this is to show that the time resolution is not very good. You can imagine that if two events occur 50 milliseconds apart here, in neuronal events occurred 50 milliseconds apart, you will not be able to separate them with a bold signal. And we'll see that this is actually playing to our advantage. So going back to our story of EEG and fMRI, we've seen the cerebrovascular coupling. The fMRI is not specific to epilepsy. In other words, if you do an fMRI on a patient, you get no specific information related to the epilepsy, unlike if you do an EEG, where you get information that is specific to epilepsy. The spatial resolution, of course, is high. The temporal resolution, as we just saw, is low. What is interesting, though, is that the fMRI is sensitive to deep brain regions. It's equally sensitive to anywhere in the brain, unlike the EEG. So what happens when we combine EEG and fMRI? This is what we get. What we do is fMRI based on EEG. So we don't reflect directly neuronal coupling. We're still going through the cerebrovascular coupling. We become specific to epilepsy because we know that we can do fMRI at the time of spikes because we have our EEG. We have high spatial resolution, low temporal resolution, and high sensitivity or sensitivity to deep brain regions. So this is really the key to why we do fMRI based on EEG, because we have these set of characteristics, and you will see how we can use them to our advantage. So this is in very brief terms how the procedure goes. We put the patient in the scanner with EEG electrodes, and we have to select patients who are likely to have interictal epileptic discharges during the time we will scan them. If they're not likely to have any spikes, then there's no point putting them in the scanner. They don't need to have many spikes, but a few spikes, we have to select them in this way. We end up with an EEG that is terribly contaminated by artifact because you're in the scanner, and this is a big problem. However, we can remove most of these EEG artifacts. We mark the spikes on the EEG, which is the key aspect of our analysis. We have to know when spikes occur during the scan. And this is the hard part. In other words, this is where the EEG is not clean because of these artifacts. We remove them, we don't completely remove them, we distort the EEG, but we have to be able to find where the spikes took place. And then we determine the brain regions that show a significant bold change at the time of these spikes. And then we superpose these voxels onto an anatomical MRI. So let's look at some examples in focal epilepsy. This is a spike on the left here, recorded outside the scanner, classical temporal lobe spike here. And this is how it looks after we've removed the artifact, recorded inside the scanner. You can see the EEG is not quite the same, but it's easy to recognize that this is a spike. And that's all I care about, is to know that at that time there is a spike. It's a little distorted, and sometimes it's more distorted than this, but it doesn't matter. I know that there is a spike. And then I do this sequence of analysis I just mentioned, and then what we get here is an activation in the left temporal region. All the pictures I will show you is left and right is right. So we have an activation here in the superficial posterior temporal neocortex, which is very limited to that region. You can see it in a few slices, nothing else anywhere. And we can presume that in this patient, this is where the generator of the spike is located. Now I'll show you an interesting example. This is a patient suspected of bitemporal epilepsy. Bitemporal spikes, quite classical in their distribution. temporal lobe type of seizure, the symptomatology, I won't go into the detail. What was strange about the patient is that absolutely no memory deficit. And of course, that's a red flag because you really expect some specific memory deficit, verbal or nonverbal, and no structural hippocampal abnormality. That, of course, occurs in temporal lobe epilepsy, but with the absence of memory deficit, it made that a bit strange. So what did we find with the EEG fMRI? Well, we have here some left temporal spikes, quite classical left temporal spikes, and where we found their activation is in the left insula, very clearly. We have a little scattered activation here and there. We have a deactivation here in blue, which I won't spend too much time on, but this is actually part of the default mode network, and this is secondary to the primary epileptic activity, but we have clearly an activation in the left insula. Right temporal spikes that you see here, the phase reversal here, activation in the right insula very clearly, and curiously enough, bitemporal spikes, which occurred rarely in this patient, activation in both insulae as well as the mesial frontal region. So this is a case which I think is quite rare of biinsular, looks like biinsular epilepsy. We haven't done anything for this patient, no question of surgery at this time, but this is to show you what you can discover with the EEG fMRI that fits very well with the clinical history of the patient, no memory deficit, temporal lobe type seizure, may have some insular specific symptomatology, but still this is not something we could have seen with any other method that I know of in this patient. This is another example, a patient with right temporal spike with polymicrogyra. You see the abnormality quite well, bilateral perisylvian polymicrogyra here, right temporal spike here. And you see what is interesting in this patient is that we have an activation that is limited to the posterior part of the polymicrogyra. Now, whether the patient can be cured by removing that, and it's not clear. We know very well that these patients don't respond very well to surgical treatment, but still it tells us that maybe this is the most epileptogenic region. Now, I come to what I think is an interesting example. This is a left temporal spike, phase reversal at F7, very classical left temporal spike here, and an activation in mesial temporal structure, very clearly. Very little bit in the pole here, but primarily in mesial temporal structure. You see it here, you see it here. So you can say, well, this is typical left temporal lobe epilepsy, and it's what we expect. And I'm going to spend a bit of time to discuss that because this is really not what we expect. Because a spike occurring in the hippocampus is not visible on the scalp EEG. This we know very well, and this is one of the things that if you're not convinced, I would really like you to become convinced. It's been demonstrated many times in many papers with combined intracerebral and scalp EEG studies that a spike in the mesial temporal structures in the hippocampus, if it occurs in that region by itself, is not visible on the scalp. So what we've seen on the scalp is not that. What we see on the scalp EEG in temporal lobe spike reflect spikes that are occurring in temporal neocortex. That's the only way we can see them. And so what we've recorded is a spike generated in the temporal neocortex. So why does such a spike result in mesial temporal bulb activation? It really should not. So this is what we think happens. A spike occurs in mesial temporal structures. It propagates to the temporal neocortex, and then it's recorded on the scalp. And the bulb activation is highest at the region where the spike originated in mesial structures, not where it propagated in the neocortex, presumably because energy consumption is highest where it was generated, and it's highest even because it propagated. That required even a stronger neuronal discharge. And this is essentially, in these few words, there is the key of what I would like to convey today, as you will see. So this is in a way similar to what we do with ictal SPECT, which reflects the region where the seizure started, not where it propagated. Even though we may do an injection after the seizure has already propagated, what we see is where it started, because where it started is where most of the blood flow takes place in the SPECT study. And this is an example here from a patient with intracerebral electrode. This is one intracerebral electrode, different contacts from mesial to lateral. And this is a mesial channels here. You see a spike occurring here at this time of the line. And then this is in the temporal neocortical contacts, in the depth electrode, but in the neocortical contacts. And you see that we have a 30 millisecond delay here. Now you remember that the bold cannot separate that. So when we use this signal here to trigger the bold, it will look anywhere in the brain for whatever happened around that time, whether it occurred 30 milliseconds later or earlier, doesn't matter. So it will find the maximum bold actually in this region because this is where the event was triggered. This is another example. Here we have a depth electrode aimed at the amygdala. We see a spike here in the amygdala at time 0, and what we record in the cortex, on the neocortical contact of the intracerebral electrode occurs with a similar delay here of 20 to 30 milliseconds. Now I'll show a little more detail of an example here where we have actually both the fMRI and the intracerebral electrode in the same patient. So this is the EEG fMRI. You see that the sharp waves in this event are bifrontal. You see them here at F3C3, F4C4. As I explained before, they're not great morphology. They're distorted by the scanner, but we know this is what it looks like in this patient, and this is what we recognized. So this is what the events look like, and this is what the fMRI, the bold fMRI looks like. We have a maximum bold in the left anterior cingulate. It's bilateral, but it's maximum on the left side here. Now subsequently to this study, and not because of this study, because this was done as a research study, the patient had intracerebral electrodes because there was a suspicion of a lesion in this region, of a focal cortical dysplasia. We see here the electrode that was placed, called LCA, left cingulate, anterior, and then another electrode here, left cingulate posterior. Patient had other electrodes. I don't have time to show all the results. But what did we find with this electrode? In the left anterior cingulate here, in the deepest contact, we have a pattern quite typical of focal cortical dysplasia, almost continuous spiking in this region. Now what happens at this time here? We have one spike, we have, as you see, continuous spiking in the superficial contacts, and this is a simultaneous EEG recording, scalp EEG recording. We don't see anything at the time of these events, of this event or this event here. Now at this time here we have a spike which propagates to the neocortical contacts, which we see in the scalp EEG with a delay of about 20 milliseconds. So this is what happened in this patient, and we triggered our fMRI from this scalp event, and we found the maximum bold in the anterior cingulate deep, not in the superficial contacts, which are the contacts generating this event. We found the maximum bold where the event originated, the neuronal event originated in the anterior cingulate. I'll show you a few more examples of a similar situation without going into so much detail. These are patients with periventricular nodular heterotopia. We have this widespread discharge here in the right temporal region. Oh, I realize you don't see my mouse. Do we have a pointer? I looked at my computer and I see my pointer. There's no pointer. Oh, that's too bad. Well, you see the right temporal discharge here, and you see the nodular heterotopia where we have the yellow arrow here in the anatomical MRI scan. And then you see at the bottom where the red arrow is, you have the fMRI activation, which corresponds exactly to the nodular heterotopia. We see it better in this picture here, where you see the nodule on the right side and the bold activation in exactly the same region. Again, the EEG that we saw was generated in the neocortex, is not generated, it originates in the nodule, spreads to the neocortex, and that's what we see in the scalp EEG. This is another example here. Again, bilateral periventricular nodular heterotopia. You see the yellow arrows on the right side show you where the nodules here, and we have bold activations bilaterally primarily on the right side in the nodule, but also on the other side a little bit in the nodule, together with some scattered deactivation. The patient has right posterior temporal burst of sharp waves that you can see on the EEG. And again, if we look more closely, we see the correspondence between the maximum bold activation and the nodule deep in the brain. This is another example here where we see on the EEG with the red arrow, you see some rapid polyspike and slow wave at the midline and at P3, at CZ and at P3. We have a depth of the sulcus focal cortical dysplasia with the green arrow shows, which we marked with a white dot in the middle there. And you can see that the bold activation is exactly in that region of the lesion, quite deep. And again, this activity that we see in the EEG is generated more superficially. This is from the recently published from the work of the group of Dr. Ikeda in Kyoto in Japan, and they looked at hypothalamic hamartoma, and they actually compared that with the SysCom. But what I want to emphasize is the presence of a bold activation in the hamartoma, even though, of course, the EEG spikes that we record are generated far away in the temporal neocortex. But this is the origin of the epileptic discharge. So, we can ask the question, can the EEG fMRI be useful in pre-surgical evaluation since it is based on the study of interictal events which are known to be relatively widespread? We know very well, it's been documented many times, that the interictal zone, the irritative zone, is quite widespread and often does not correspond well with the seizure onset zone. So can we use these findings in surgical results? And we did a study here a few years ago. This is the work of Dongmei An from Chengdu. We took patients having an EEG fMRI study, having had surgery and at least a year follow-up. We co-registered the post-op MRI with the pre-op bold so that we can see the region that was resected and compare it to the region where we had the bold activation. And we divided our patients in 4 groups according to the concordance between the bold activation and the subsequent surgery. Now you have to realize that these patients were not operated as a result of the bold study. This is a retrospective study and was done as research at that time. In 10 patients, we had full concordance between the bold activation and the region that was resected. In 9, we have a relatively good concordance. In 5, we had a partial concordance, just a little bit of overlap. And then in 11 patients, we have a total discordance. The bold activation is here and the surgery is there. And so what happens to these patients? You can see here in the first group, 7 are seizure-free. And what is particularly important is the last group, where only 1 patient is seizure-free out of that group. And so what that tells us really is that it's not a good idea to leave behind the region of bold activation because the chance of success, of surgical success, is very poor in this patient. And it's much higher if that region is removed. For the groups that are in between, we have mixed results. I'll show you a couple of examples of these. This is a patient who had a normal MRI, had depth electrodes, and following the depth electrode investigation, he's part of the first group, the patient became seizure-free. You can see on the right that there is almost perfect overlap between the region of bold, which was done, of course, pre-surgically, and the surgical cavity, which is in fact largely hidden by the bold. They almost perfectly overlap. And this is another case of the patient who was not seizure-free. Again, a patient with left polymicrogyra who had depth electrodes to try to localize the part of the polymicrogyra that was epileptogenic. And the surgical cavity you see is in the orbitofrontal region, inferior frontal region here. And you can see that the bold response is bilateral, but the maximum is clearly on the left above the surgical resection. So that region had not been explored with depth electrodes, and the region where we thought the seizures were starting from was removed. Of course, the problem there, and I will come back to that, the big issue is that with depth electrodes you can cover only a very small fraction of the brain. And that is the reason here for this failure. So, to answer the question I asked earlier, can EEG fMRI be useful in pre-surgical evaluation since it's based on the study of interictal events, which are known to be widespread? We just demonstrated that yes, it is useful. And then you can ask the question, why is it useful if it in fact represents such widespread activity? And this is where I want to introduce the concept of the spike onset zone. Now, we've heard before many times of these many zones, the irritative zone or the spiking zone, which is usually quite large, the epileptogenic lesion, which is usually smaller if it exists, the seizure onset zone, which partially overlaps the lesion and usually is inside the irritative zone, the epileptogenic zone, which is a mythical kind of zone because we don't really know what it is, but it's what you need to remove to make the patient seizure-free. And what I want to introduce, as I said, is the spike onset zone. Now, we know that the irritative zone is quite large, and the question is, do we have a spike onset zone? This represents a widespread irritative zone. We record spikes a little bit everywhere. And the question we ask, did they start there? Is this the spike onset zone? Or is it there? Or is it there? How do we know? We can't record everywhere. So this is where the EEG fMRI method starts. We pick a spike anywhere where we can record it. In this case, we record it on the scalp. And then we do an EEG fMRI, and what we find, I claim, is the spike onset zone. This is the region of maximum bold. So, assuming that the spike onset zone exists and it's represented by the maximum bold, why is it interesting? Why is it why is it good at predicting surgical outcome? And I think it's because it corresponds to the seizure onset zone. And this is what I would like to try to demonstrate to you. This is a study that was done recently and just accepted for publication. This is done by Dr. Koo, who's been working with us. What we did is we took patients who had an EEG fMRI studied, followed by electrode implantation. And we assessed the correspondence between the region of maximum bold, the spike onset zone, what I call the spike onset zone, and the seizure onset zone as recorded by intracerebral EEG. We superimposed the pre-implantation bold map and the electrode positions, and we defined the concordance if the contact of seizure onset are within 2 centimeters of the peak bold. I'll show you an example. This is how we evaluated the concordance between the bold response and the seizure onset zone. This is a patient's typical bold response as a maximum here in the left temporal region, and some scattered lesser activation and some few deactivations here. We first co-register this image with the depth electrode, as we see here. You can see here some of the depth electrode tracks in the left temporal and right temporal region. The patient had actually quite a few more electrodes than this. Then we define an area of 2 centimeter radius around the maximum bold, and we find that the electrodes involved during the seizure onset are within that circle. So then we say we have a concordant finding. And if not, then we will say we have a discordant finding. We did not study patients in whom the maximum bold did not have any electrode next to it, because we can't say anything about that. So we only studied patients in whom there was an electrode in the vicinity of the maximum bold. This is an example. This is complicated, and especially with that pointer here, it's difficult to explain, but you can see the maximum bold is in this region here. Actually, you can see me here. This is the region of the maximum bold, and we have depth electrode in multiple regions. You see them here in Heschel's gyrus, in SMS, in the supplementary motor inferior and superior area, and the cingulate gyrus in various regions. But we will concentrate on the electrodes, that are near the maximum bold response here. And in red, we see the contacts of the depth electrode that are involved at seizure onset, and in green that are not involved at seizure onset. And you see in the EEG the onset of the seizure in the left posterior insular cortex and in the left Heschel gyrus. This is where the onset of the seizures are. This is exactly where we have our maximum bold. So, overall, in these 31 patients that we studied, 77% of them, the spike onset zone, the maximum bold, correspond to the seizure onset zone. And this is the breakdown of these patients. We have at the top all patients, 24 were concordant and 7 were discordant. Non-lesional, this is broken down into non-lesional, lesional, focal cortical dysplasia, and nodular heterotopia. You can see that the concordance level is about the same in these different groups. It's a little bit lower in non-lesional patients. Now, I want to emphasize that the discordant results are not necessarily erroneous results. Okay, they're discordant in the sense that we found the seizure onset not where we had a maximum bold, but because of the minimal coverage of depth electrode, we're not really 100% sure of where the seizure really starts. The seizure may start where we don't have an electrode, someplace that may be not too far from our bold maximum. Excuse me. Now, what was interesting is in about a third of our patients who have a specific bold pattern that I will describe, the spike onset zone determines with almost certainty, that is with 93% probability, the seizure onset zone. So in particular situations, which I will show you, we can say if this pattern of bold response occurs, we have a probability of 93% that this is the seizure onset zone. And the pattern is the following. Here we represent the bold responses in all our patients on two axes. At the bottom is the absolute t-value, in other words, how high is the maximum bold response. And on the left side in the vertical axis is the difference between the maximum t-value and the second highest t-value. So what you can see is if this difference is high, and which means we're at the top of the screen, and if the bottom one is to the right, which means we have a high t-value, and the second highest t-value is low, which means we have an isolated peak bold response, or almost isolated, then in that group we have a 93% probability of finding the seizure onset zone, which is very high. So coming back to what we were, the seizure onset zone is very often, the spike onset zone, which I wrote with a small s here, is very often the same as the seizure onset zone, hence its value. What are the consequences for implantation of electrode and surgery? If it is decided to implant electrodes in the patient, the maximum bold provides a very strong hypothesis regarding the seizure onset zone. This is important because intracranial electrodes explore a small fraction of the cortex. We need strong hypotheses. We need to know where to put the electrodes. As I've shown you in one example, if you put them in the wrong place, if you don't hit the right target, you're going to find the seizure onset. It just won't be where it really started. So you have to have a strong hypothesis. Either there is a lesion or there is a potential lesion, or you have, in this case, a maximum bold. And what we're saying is that the maximum bold is a very strong hypothesis. And I want to show you a picture which I saw once, which struck me to illustrate how intracranial electrodes cover a small part of the brain. This is a large subdural grid that you may think has a good coverage of the brain. However, if you blow up the brain, this is where your electrodes are actually, and I presented here on the blown up brain where the sulci are shown on the surface as well. You can see how poor a coverage of the cortex, your grid, which looks like it covers a large part of the brain, is actually doing. And this is what I mean. And with intracerebral electrodes, it's the same thing. The coverage is very sparse, so we need to know where to put electrodes. And in patients with very high probability that we have found the seizure onset zone with the bold, that group of patients that I showed before, it may not be necessary to implant electrodes. I mean, why do we implant electrodes? The only result of electrode implantation is to find where the seizure onset is. And if we can demonstrate that we're almost sure to have found where the seizure onset is, then you can really ask the question, should I implant electrodes in these patients? Now, to conclude, maybe we'll spend a little time on epilepsy-related networks. This is maybe less relevant for the clinical care, but I think it's a very interesting aspect that we are able to study with EEG fMRI. This is the default mode network. I'm sure you're familiar with this network. It's been discovered, and this is in healthy subjects. It's been discovered that these regions of the brain, the posterior cingulate, the anterior frontal region, a little bit the temporal region, the mesial temporal region, these regions seem to act in concert at the time where you're just lying in a scanner and doing nothing, being awake and your eyes closed or your eyes open, but doing nothing specific. These parts of the brains are active at that time. As soon as you start to do something specific, then they become deactivated. So this, I'm sure you're familiar with this network. What we found quite a few years ago now is that in patients with idiopathic generalized epilepsy, we found that this network was deactivated at the time of spike and wave discharges. What you see here is the result of a group analysis of 13 patients with idiopathic generalized epilepsy. And you see that almost the regions that are deactivated correspond exactly to the regions of the default mode network. We hypothesized at the time that because this network is involved in maintaining attention, in allowing you to monitor your environment when you're awake, its deactivation at the time of spike and wave discharges may reflect the inability of the patient to actually monitor their environment at the time of spike and wave discharges. In other words, their disconnection from the world, their unawareness, if you like, lack of awareness during their discharge. This is a hypothesis. Other people, or it's also possible that in fact it's the deactivation of this network that causes the spike and wave discharge. And because of the lack of temporal resolution in the fMRI, we don't really know which is first. Now, what is interesting is that this network is also deactivated with focal spikes. It's not as deactivated as dramatically, but you can see here a patient with left temporal spike, left temporal activation, but you can see that the posterior cingulate and the anterior frontal left region and these are parts of the default mode network, are clearly deactivated. So it was a very surprising finding, because what it tells us is that even though this is a very focal spike, it looks like it may have an effect on this network that allows you to monitor your environment. So these at quite a distance from the focus. By what mechanism, we don't know, but it looks like this network may be deactivated. This is another example here, right temporal spike, a clear activation in right temporal region, mesial and neocortical in this case, and again the posterior cingulate, which is the most prominent node of the default mode network, is deactivated. This is a group analysis of patients at the top of patients with temporal lobe epilepsy, at the bottom patients with frontal lobe epilepsy, and ignoring the activation. I should have said that all the patients are flipped so that they all have left-sided lesions. You can see that in the temporal lobe epilepsy you have the posterior cingulate and the contralateral parietal parts of the default mode network are deactivated. And in the frontal lobe epilepsy, even though they have frontal lobe epilepsy in many different parts of the frontal lobe, there is a small deactivation in the contralateral parietal region. So, and very similar results were obtained by the group of Fernando Sendes, Dr. Cohen in Brazil. It's a little harder to see here, but these are patients on the left side with mesial temporal lobe epilepsy with hippocampal sclerosis, and on the right side mesial temporal lobe epilepsy with a normal hippocampus. And you can see in both groups that you have some deactivations. These are group analysis, almost exactly the same regions that you saw in our previous study. So again, this mesial temporal lobe discharges, this is based on mesial temporal lobe discharges, cause deactivation in the default mode network at quite a distance from the spikes. Now, I want to spend a minute for a recent study that we're just completed very recently, studying epileptic networks during interictal spikes. We investigated, rather than the maximum bold cluster, which is what I told you about so far, which tells you where the spike onset zone is, you've noticed that we had scattered activation sometimes in some patients. And we looked at these scattered clusters of activation resulting from presumably focal spikes. And we did a correlation between the spikes inside and outside the clusters in a subsequent SEEG, stereo EEG study. So what we found is we had a high correlation between spikes if they were within bold clusters. Now, realize the bold clusters were recorded at one time. The SEEG was done a year or 2 years later, but we co-registered, just like we did with the other study, we co-registered the activity, the depth electrodes with the bold clusters. And if the spikes occurred in both two regions that were part of bold clusters, then we had a high correlation. And if we correlated the spikes, some within a cluster and some outside a cluster, or completely outside a cluster, then we had a low correlation. So what we conclude is that bold clusters therefore represent a network of epileptic activity. At the time of a scalp spike, a widespread network of epileptic discharge is active, and these bold responses tell us about this scattered network. And it has been discussed very often. People say we have a scattered bold activation that represents an epileptic network. But there was no demonstration that this is actually epileptic activity in these different regions. So, what do we conclude about the use of EEG fMRI in focal epilepsy? Studying fMRI at the time of epileptic discharges is a non-invasive examination of the whole brain, which applies equally well to superficial and deep generators as long as the discharge is present on the scalp EEG. But discharges are often present as a result of propagation. And here I will make a little aside, which comes from a very recent study, which is not published yet, where we looked at, we often say, I'm sure all of you say, oh, the patient doesn't show any spikes because the lesion is very deep, so we don't see any spikes on the scalp. This is an explanation for why we don't see scalp spikes. So we actually studied two groups of patients, some with deep lesion and some with superficial lesions. And we found that they're equally likely to have spikes on the scalp. So the depth of the lesion is actually not a factor in the presence of scalp spikes. And that comes most likely from the fact, as I mentioned before, that spikes propagate. So either the spikes propagate, and it doesn't matter that they start deep, you will see them on the surface, or they don't propagate and then they're very local and then you just don't see them on the scalp. So very often we see scalp spikes even in deep lesions, as I've shown in several examples here. This results in a bold response that is a good predictor of seizure surgery outcome and of the region of seizure onset. This may reflect the fact that the maximum bold response represents the region where interictal discharges are generated. And you have to be careful here not to confuse volume conduction with neuronal conduction. When I say they're generated, I mean they're generated neuronally and then they propagate to neocortical regions neuronally. This is not volume conduction we're talking about. And this concept led us to the concept of the spike onset zone, a zone that may largely overlap the seizure onset zone. The traditional irritative zone is a poor marker of seizure onset zone because it includes the spike onset zone and the regions of spread. So this really confirms the old idea that the irritative zone is not a good marker. What you have to find is the spike onset zone. fMRI based on EEG reveals relative deactivation of the resting state network, primarily the default mode network, around the time of epileptic discharges. And when multiple regions are active on bold at the time of spike discharges, these regions may reflect the involvement of an interictal epileptic network, reflecting the ability of spikes to spread. So we come back to the same idea. Now, as I've mentioned at the beginning, combining EEG and fMRI in focal epilepsy remains a difficult method to put in place for routine clinical practice. The interpretation of the EEG recorded inside the scanner is challenging because of its poor quality. And this is really the key difficulty in the more widespread use of this method. I was discussing this with Dr. Kubesi earlier. I think that the method itself is not expensive. You can use a standard fMRI, MRI machine. The EEG, you need to record to use special EEG amplifiers that are MR compatible. These are not particularly expensive. You can use 32 channels of EEG. And so the equipment you need is not complicated. The procedure is not very complicated. You put the electrodes, you put the patient in the scanner. You have to be very careful to have the patient well immobilized so that they don't move during the scanner, so that they're comfortable. Usually they'll fall asleep, which is very nice because that activates their spiking activity. But then you have an EEG that is full of artifact, and because of the EPI, because of the scanning itself, because of other artifact, the ballistocardiogram is a very serious artifact, and you have to work to remove this artifact so that you can see the EEG and mark the spikes. And that's the hardest part. There are no absolute standard method to remove the artifacts. There are several methods that have been out. There are always new methods that are being developed. And you end up with an EEG which is not great in quality, and you have to carefully mark where the spikes take place. This is the key to obtaining a good study, is to mark where the spikes take place. After that, the analysis is done in a relatively standard way. So to finish, I want to mark our contributors, thank our contributors to this work, particularly Dr. François Dubois, who is the epileptologist with whom I have been working for many years in Montreal, and without whom this work would not be possible. I'm not a clinician myself, but he is now dependent on this method for the investigation of many patients. And other fellows that have worked here, particularly on this project, Francesca Pitau, Firas Faoum, Natasha Zazubovitz is a research assistant, Louise Tiver Dongmean, Christophe Grova is a young colleague working with me on these, and Huiming Ku, who has done the study I just mentioned about the combined intracerebral and fMRI analysis. So these are everything you need to know about claiming CME credits for this activity. I think there is a code here that is key that you have to see. And thank you for your attention.
Thank you very much for the beautiful and inspiring talk, as always. We have a couple of questions here, and then I'll ask if people in the room have other questions. First, do you think it's good practice if you have a patient with a single ictal discharge distribution but multiple interictal populations on scalp, to pick the spike population that most corresponds with the ictal discharge and then see which bolt signal corresponds to that in an attempt to identify the spike onset zone of that particular spike population?
Well, what we do in the patient who has multiple interictal patterns is, if all of them are represented during the scan, is to analyze them separately. In other words, we mark each one, but we mark them as event type 1, event type 2, event type 3, and then we do a combined analysis of all these events, and then we can find what is the bolt response to each one. And then we presumably have found the spike onset zone for each of these events. And I think now, if the patient has a single seizure generator, then presumably one of them will correspond to the single seizure generator. But I think that in this case I would be a little wary. I would be a little concerned, you know, if one of them looks like it corresponds to the seizure onset generator, but then there's another one somewhere else and another one somewhere else, then you know, this patient may not be so simple. And there may be that you haven't recorded other seizures the patient may have. I don't know, but I would certainly take these into consideration. What we have relatively often, this is something we're looking at now more systematically, as you know very well, you often have patients who will have a spike pattern that is sometimes more focal and sometimes more widespread, even though it seems to have a similar maximum. And we found that, and we analyzed them in these patients, we analyzed these two patterns separately. You may have a temporal pattern that sometimes becomes frontotemporal, for instance. We analyzed them as two event types separately, and very often we find that the maximum bold response is the same, but the extent of the bold response is different.
Have you looked at high frequency oscillations in by depth electrode in areas that that were found that were the spike onset zones?
Well, I give you the answer next year. This is currently under progress. So because we have the co-registration of the depth electrode with our bold response that we have done for the study of the correspondence with the seizure onset, we are able to do the relationship, to study the relationship with the high-frequency oscillations. And this is a work that Karina Gonzalez in my lab is currently under doing. But I can say that we've looked at high-frequency oscillation in scalp EEG in these patients. Now, you know that you can record high-frequency oscillation in the scalp EEG. Not very well, and they're not very frequent, but it's very clear that you can record them. You can record ripple activity. And mostly they occur around the time of spikes. So what we wanted to do was to compare the patients who had spikes with ripples and the patient who had spikes without ripples and see if their bold activation was different. Now, this turned out to be a very difficult study because you can see the ripple in a good scalp EEG, but you cannot see the ripples in an EEG recorded in the fMRI. Because in the fMRI, in order to remove the artifact, you have to use some low-pass filtering, and you remove your ripples. But Firas Faoum, who I just mentioned here, I showed in the picture, had a very good idea, and what we did is that we took patients who had frequent ripples with their spikes outside the scanner, and patients who never had ripples with their spike outside the scanner. And then we put them in the scanner, and we just assumed that their spikes would do the same thing. We can't see it, but we just assume. So we put a group of patients who had spikes probably with ripples and patients who had spikes probably without ripples. And we compared the two groups. And I was convinced that we would find more intense bold response in those who had ripples. And we didn't. But what we found, and I'm still not clear on why, but there was a very strong finding, this was published a few years ago, was the patients who had ripples were most likely to show a thalamic activation at the same time as the focal neocortical activation. And why that is, I leave it up to you to give me some idea. But my only hypothesis was that, it was an ipsilateral thalamic activation, which we see sometimes. I didn't mention it here, but we see sometimes in some patients. And this was present in those who had ripples. And to me, that is an indication that thalamocortical mechanism probably play an important role in the triggering of even focal spikes, something we don't think about very often. But so that's what we found.
If I remember correctly, there was also some thalamic activation in the absence seizure network.
Oh, in the absence seizure, this was very clear. We had very clearly, in most patients during the spike and wave, we had thalamic activation. I only showed the deactivation here because of the default mode. But we had this was the clearest activation in the absence seizure was the thalamic activation, yeah.
We have one last question here. The duration between the spike and the maximum bold signal, is it different with different seizure foci and different patients?
So I showed you this hemodynamic response function that is typically used for pretty much all fMRI study, where it shows a peak about 5 or 6 seconds after the neuronal activation. In order to do the fMRI analysis, you have to use this hemodynamic response function. So you have to prejudge of this time difference, if you like. What we did over the years is develop a method where we actually include several hemodynamic response functions. One that peaks at 3 seconds, one that peaks at 5 seconds, one that peaks at 7 seconds, and one that peaks at 9 seconds. And we analyze all of them together and then we take the one that gives us the highest t-value. And in some patients it's earlier, in some patients it's later. We haven't found a systematic difference. In fact, we actually looked at different patients with lesions in different regions, with different types of pathology, and we haven't found a systematic difference. So I don't really know why that is, why there is a difference.
Well, thank you. Any questions? Yes.
Yes, thanks for the talk. Yeah, so I have a couple of questions. The first is like in clinical, like we.
I'm sorry, could you speak a little louder?
Oh, okay, so clinically, like we see quite often, like those we call like the bisynchrony, like the bilateral frontal, those spikes, and we know for sure or pretty sure like those are focal tissue from one side, but it's just from the scalp EEG. It's very hard to tell, and also from the ictal onset, sometimes hard to tell. And I'm curious, when you saw those in like the functional MRI, do you kind of always see the lateralization in those cases?
Yeah. We studied this quite a few years ago, and I've published one paper about this. But and we've seen that again and again. That in patients with presumably focal epilepsy or partial focal epilepsy and bilateral spike and wave, that we have usually a widespread or often a widespread bold response, but very clearly with a predominance and often a very focal accentuation. So that is very helpful in localizing the focus. We actually did another study related to this patient group, which I think was interesting. We tried to see whether there were particular frontal regions that were more likely to lead to secondary bilateral synchrony. In other words, whether the maximum was like, if it was mesial frontal, was it more likely that we would have secondary bilateral discharges, or if it was lateral, we would be less likely. And we didn't find that. In other words, it doesn't seem to be a particular frontal region. Unlike what has been said before, there doesn't seem to be a particular frontal region that is likely to lead to widespread discharges. And my interpretation at this time is that you really have a widespread region of abnormality with a focal accentuation. It's not just like you have a focus and some spread to normal brain regions.
Okay. Yeah. My second question is like, have you ever seen like when you see the spikes and then you look at the bold signal, and I'm just curious, sometimes we miss the spikes from the scalp EEG. So for all those like without spikes, those period, if you look at like the bold signal, are those will be all normal? Or if you see, I'm just curious, some like a bold signal without any spikes, you see it in the EEG?
Well, you see, this analysis is requires the presence of an event. Mm-hmm. In other words, when we do the analysis, I didn't show the detail of the analysis method, but it's called an event-based fMRI analysis. And what it requires is that you have events, and what you do is you build a model. This model, it's a model hemodynamic response. So you say, if there is no spike, I have no response. My model is flat. If there is a spike at this time, then there must be this hemodynamic response function I showed you before, which peaks at 6 seconds. So I put this in my model. Then I said there's no spike, it's flat. Then there's another spike I create. So this is my model. Okay. And then I correlate this model with the time course of every voxel in the brain. If it looks like this model, then it must generate the spike. If it doesn't look like this model, it doesn't generate the spike. But if I don't have timings, sure, I can't do anything. Sure. This is why I'm saying if the patient doesn't have spikes. You had a question in the back there?
Yeah,
Some.
So, what happens if you just do this and you analyze without spikes? Is that possible? Can you still find a bold signal?
Well, this is the question that was given before. Yeah, I mean, I should say, if we go into a little bit of detail, some people have tried, and we have tried. I mean, I talked a few years ago when I was more optimistic about this. I talked about EEG fMRI without EEG. In other words, trying to find in the fMRI signal itself the equivalent of a spike. you can say, I'm going to look at my bolt signal just like I look at my EEG, and maybe there are times where there is something occurs, and that's because of a spike that occurred that I can't see in my EEG, or even I don't have an EEG. So we looked inside the bolt signal to see if there were fluctuations that looked like there could be spikes, and to see whether we could actually detect epileptic events in the bolt signal if nothing happens in the EEG, or even if you don't record the EEG. And we found in a small number of patients that we were able to find something, and it corresponded to, we actually had an EEG. I mean, we could do this analysis without looking at the EEG, even though we had it. And so we found that in some cases it did correspond, but altogether it was not reliable enough. Essentially, the problem is the signal-to-noise ratio of the bolt signal is very low. So if you just look, you will find all kinds of fluctuations, and you're never sure, are these generated by spike, or is it just because some spontaneous fluctuation is taking place in the default mode network, the patient is thinking about something, they're looking at something, whatever, and you don't have the specificity, which is what I mentioned at the very beginning, that you have from the EEG. So you find some changes, and then you say, is it the patient spike, or is it some random event? And this is essentially the problem we're left with. Occasionally you're right, but you don't know when you're right, and so it's just not reliable enough at this time.
So, and then sort of as a follow-up to that, what if you did the same sort of thing that we do, which is come up with the researchable hypothesis today, and then looked at what goes here, here, and here, and you just looked at specific areas based upon your hypothesis. Have you ever tried to do that?
Well, I haven't tried, and I should say that I don't really like the idea. I think that it's unfortunately with the depth electrode you can confirm your hypothesis and be wrong. In other words, I don't want to further prejudice my analysis. I think that I could look in these regions and maybe find a bold response, but maybe it's not the maximum bold response. Maybe it's a region of spread, and I don't look at other regions. So actually what I like about this method is that I'm looking everywhere in the brain without prejudice, and I discover something that is new. If I'm going to look at say, well, some people have said, for instance, you can use the EEG source analysis. Find where your EEG sources are doing, large EEG array and doing some source analysis, dipole or otherwise, and then you can say, okay, you find the source there, go look at your bolt signal there. Well, I'm going to find the bolt signal there, but what is it going to tell me? What I want is an independent measure that may confirm or infirm what I have found with one particular method. If I just confirm my prejudice, it's not particularly helpful. So I would actually rather stay away from doing that.
Great, thank you.
Thank you.